Welcome back to this series of videos in which I go through the progress check FRQs for all the units that are going to be on the AP exam. Right now we are wading through Unit 5 and looking at question 2. This video should be refreshingly short compared to the last one because question 1 was a doozy. I'm sure I'm sure you noticed that if you watched the video. And this one, hopefully, we covered it a lot in class, so hopefully you just breezed right through this one so you don't even have to watch it. But if you did run into trouble, I can go over this real quick for you. So again, refreshingly short setup here. So primary spermatocytes are diploid 2-end cells with all of the organelles typically found in eukaryotic animal cells. A representation of spermatogenesis from a primary spermatocyte with 6 chromosomes is shown in figure 1. Okay, so here's our primary spermatocyte. Here's meiosis 1, and here each one of these secondary spermatocytes undergoes an additional division, which is meiosis 2, and that gives you 4 haploid cells. Now, if you remember, what diploid means basically is it's got 3 chromosomes here, and it's got 2 copies of each. One from each parent. So, mom, dad, mom, dad, mom, dad. And then when it undergoes meiosis 1, that's where it goes from being diploid to being haploid, right? Now, these only have 1 copy, right? They have 1 copy of each, and it doesn't necessarily mean mom, 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 dad, dad, dad. It's unlikely to go that way. It's possible. There's like a 1/8 chance if you have 1 in 8 chance of if you have 3 chromosomes, but it's unlikely. And then what you're not seeing here in this diagram, because it looks like these are replicating, they're not at this point. These are going to be made of 2 sister chromatids, which is what then is going to separate into these 4 haploid cells here after meiosis 2. So that's a little bit unclear from the diagram, but I wanted to kind of re-mention that. Okay, so let's see what they want us to do here. Okay, so describe the process of meiosis that ensures both maternal and paternal chromosomes are passed on to each spermatozoan. Great. That is going to be what we kind of just talked about, our law of independent assortment. We did a lot of stuff with this in class. And that just means basically in metaphase 1, in metaphase 1 of meiosis, chromosomes line up independently of one another. You guys flipped a lot of coins modeling this independently, right? You guys flipped a lot of coins modeling this in class, and you saw that just because you got you flipped heads and you got mom's chromosome 1, it doesn't necessarily mean that you're going to get heads for chromosome 2. You can easily get tails and inherit dads. So that kind of ensures that maternal and paternal chromosomes are passed to each spermatozoan. Okay, so B, explain why the genetic content of individual chromosomes in a spermatozoan most likely differs from the genetic content of individual chromosomes in the primary spermatocyte. Okay, so basically, why are these different individual chromosomes in a spermatozoan? And that's basically this here. So why is this chromosome different from this? Well, the answer is that something happens here in meiosis 1 that I'm sure you guys will remember because you've definitely, again, modeled it a bunch of times. So, sure, there's independent assortment in meiosis 1, but there is also crossing over. So when these line up right kind of before, as this is getting ready to undergo meiosis 1, say these are chromosome 1, they're going to link up, they're going to synapse, and they're going to exchange bits, random bits of DNA with each other just at the ends, basically. And then so that way, even if you have a 1 in that 1 in 8 chance where you get dad, dad, dad here, you don't really get all of dad's DNA, because this chromosome will have a certain piece of mom's, this will have a piece of mom's, that will have a piece of mom's. And it's just another way that genetic diversity is increased in a species. So we're going to say crossing over occurs in meiosis 1, where, homologous chromosomes, if you must use the proper terminology, where homologous chromosomes exchange DNA, right? Sections. Cool. All right. So, homologous pairs of chromosomes randomly segregate. The law of independent assortment. Great. The response indicates a crossing over / recombination occurs between chromatids of homologous chromosomes during prophase of meiosis 1. Great. Okay. And then C, in some instances, meiosis of a primary spermatocyte with 6 chromosomes here results in 2 spermatozoa that contain 4 chromosomes and 2 that contain 2. Predict the most likely cause. Well, if you've got, 4 and 4, so if you had something that looked like this, right? If you had something that looked like that, there's 4. And then here, you actually were missing one of these, right? So if you're missing one of these, then it's the same one, whatever. But yeah, this would have to be gone too, right? Like, because these, remember, are going to come from, these are just sister chromatids that are going to split there. So what would have to have happened is you would have to have something called nondisjunction. You would have to have a situation where things didn't go right in meiosis 1. Things did not go right in meiosis 1. And so, yeah, you would have to have a non-disjunction event there. Non-disjunction event in meiosis 1, right? Great. And then, so you would have to have 4 here, you would have to have 4 there, which then would split into, the sister chromatids would split, 4 there, 4 there, 2 there, 2 there. And then a student claims that if the animal producing the spermatozoa has a mutation in a mitochondrial gene, the probability that any offspring will inherit the mutation is 0. Provide evidence to support this claim. Well, as you all know, mitochondrial genes are only inherited through the mother, right? So mitochondrial genes, mitochondria basically not present in spermatozoa. They're not present in sperm cells. All mitochondrial, I'm going to say mDNA, is inherited matrilineally through the mother. Okay. All right. So, this response indicates that the most likely cause is non-disjunction of one pair of homologous chromosomes during meiosis 1. And, okay, the response indicates that spermatozoa contain no mitochondria. All mitochondria in an organism are derived from the ovum. All right. Well, thank you for watching. I've got to go cook dinner so that the source of my children's mitochondrial DNA doesn't get any madder at me than she is right now. So,